Sleep and dreaming produce electrical patterns in the cerebrum. A dominant feature of human behavior is the daily cycle of sleep and waking. All birds and mammals, and probably all other vertebrates, sleep. We spend 1/3 of our lives sleeping, yet we do not know why or how. We do know, however, that we need to sleep. Loss of sleep impairs alertness and performance. Most people in our society, certainly most college students, are chronically sleep deprived. Large numbers of accidents and serious mistakes that endanger lives can be attributed to impaired alertness due to sleep loss. Yet insomnia, difficulty in falling or staying asleep, is one of the most common medical complaints. The electroencephalogram, a common tool of sleep researchers is the electroencephalogram EEG. To record an EEG, electrodes are placed at different locations on the scalp, and changes in the electric potential differences between electrodes are recorded over time. These electric potential differences reflect the electrical activity of the neurons in the brain regions under the electrodes, primarily regions of the cerebral cortex. Hence, writing on a moving chart are used to record the patterns of these differences. Figure 12a, b. Usually, the electrical activity of one or more skeletal muscles once also recorded on the chart. This record is called an electromyogram (EMG). EEG and EMG patterns reveal the transition from being awake to being asleep. They also reveal that there are different states of sleep. In mammals other than humans, two major sleep states are easily distinguished. They are slow wave sleep and rapid eye movement (REM) sleep. In humans, we characterize sleep states as non-REM sleep and REM sleep. 12 patterns of electrical activity in the cerebral cortex characterize stages of sleep. Electrical activity in the cerebral cortex is detected by electrodes placed on the scalp and recorded on moving chart paper by polygraph. B. The resulting record is an electroencephalogram EEG. C. During the night, humans cycle through different stages of sleep. At awake, W. Tom Mount. Human non-REM sleep is divided into 4 stages. Only the 2 deepest stages are considered true slow wave sleep. When a person falls asleep at night, the first sleep state entered is non-REM sleep, which progresses from stage 1 to stage 4. Stages 3 and 4 are deep, restorative, slow wave sleep. This first episode of non-REM sleep is followed by an episode of REM sleep. Throughout the night, we experience 4 or 5 cycles of non-REM and REM sleep. Figure 12 cents. About 80% of our sleep is non-REM sleep, and 20% is REM sleep. We have vivid dreams and nightmares during REM sleep, which gets its name from the jerky movements of the eyeballs that occur during this state. The most remarkable feature of REM sleep is that inhibitory commands from the brain almost completely paralyze the skeletal muscles. Occasional muscle twitches break through the paralysis, as can be seen in a dog that appears to be trying to run in its sleep. If you look closely at a sleeping dog when its legs and paws are twitching, you will be able to see the rapid eye movements as well. Probably the function of muscle paralysis during REM sleep is to prevent the acting out of dreams. Sleepwalking occurs during non-REM sleep. 20:01234567 time hours. Cellular changes during sleep. There are striking neurophysiological differences between non-REM and REM sleep. Non-REM sleep is characterized by a decrease in the responsiveness of neurons in the thalamus and cerebral cortex. Remember that neurons have a negative resting membrane potential and a threshold for firing action potentials. Usually the resting potential is below the threshold potential, so the neuron is not firing. When synaptic input causes the membrane potential to become less negative, depolarized, the cell can reach threshold and fire action potentials. During waking, several nuclei in the brainstem are continuously active. Many axons from these nuclei extend to the thalamus and the cerebral cortex, where the neurotransmitters acetylcholine, norepinephrine, and serotonin they release are generally depolarizing. Therefore, these broadly distributed neurotransmitters keep the resting potential of the neurons of the thalamus and cortex close to threshold and sensitive to synaptic inputs, thereby maintaining waking. With the onset of sleep, activity in these brainstem nuclei decreases, and their axon terminals release less neurotransmitter. With the withdrawal of the depolarizing neurotransmitters, the resting potentials of the cells of the thalamus and cortex. 58 Institute for Advanced Studies, Jodhpur. Unit 7 System Physiology Animal. Become more negative, hyperpolarized, and the cells are less sensitive to synaptic input. Their processing of information is inhibited, and consciousness is lost. An interesting neuronal event happens as a result of this hyperpolarization. The cells begin to fire action potentials in bursts. The synchronization of these bursts over broad areas of cerebral cortex results in the EEG slow wave pattern that characterizes non-REM sleep. Studies of neurons of the thalamus and the cortex have shown that their hyperpolarization during non-REM sleep is due to increased opening of K channels, and that the bursting is due to CA channels whose inactivation gates close rapidly and require hyperpolarization to be reopened. We can therefore explain the EEG pattern of non-REM sleep in terms of the properties of neurons and ion channels. At the transition from non-REM to REM sleep, dramatic changes occur. Some of the brainstem nuclei that were inactive during non-REM sleep become active again, causing a general depolarization of cortical neurons. Thus, the synchronized bursts of firing cease, and the EEG resembles that of the waking brain. Because the resting potentials of the neurons return to near threshold levels, the cortex can process information, and vivid dreams occur. However, the brain inhibits both afferent and efferent pathways. Therefore, the activity in the cortex is unconstrained by its usual sources of information. One example of the effect of this loss of motor output and sensory input is the frequently reported dream in which a person is trying to run but cannot move. We do not know the function of REM sleep, but since a wide variety of mammals have about the same percentage of total sleep time that is REM sleep, it is probably a rather basic cellular function. A prominent hypothesis about the functions of sleep is that it is essential for the maintenance and repair of neuronal connections and for the neuronal changes that are involved in learning and memory. However, evidence for such functions is still meager. Some learning and memory can be localized to specific brain areas. Learning is a modification of behavior by experience. Memory wants the ability of the nervous system to retain what is learned and what is experienced. Even very simple animals can learn and remember, but these two abilities are most highly developed in humans. Consider the amount of information associated with learning a language. The capacity of memory and the rate at which items can be retrieved are remarkable features of the human nervous system. Learning. Learning that leads to long-term memory and modification of behavior must involve long-lasting synaptic changes. Synaptic changes that last for weeks have been observed. See figure 17. High-frequency electrical stimulation of certain identifiable circuits of the mammalian hippocampus makes them more sensitive to subsequent stimulation. This phenomenon is called long-term potentiation (LTP). In contrast, continuous, repetitive, low-level stimulation of these hippocampal circuits reduces their responsiveness, a phenomenon that has been called long-term depression limited. LTP and limited have been demonstrated in circuits other than hippocampal circuits, and they may be fundamental cellular or molecular mechanisms involved in learning and memory. Question: where is a conditioned reflex stored in the brain? Method: when the eye blinks, a thread attached to the third eyelid. Trigger EREST. Movement. Measuring device. A speaker delivers a sound to the rabbit. Speaker. Airnozelet. Conclusion: a small and specific portion of the cerebellum is necessary for leaning this conditioned reflex. 13. The conditioned eye blink reflex depends on a cerebellar circuit. A small and specific area of the cerebellum is necessary for a rabbit to form a conditioned reflex. A form of learning that is widespread among animal species. One is associative learning, in which two unrelated stimuli become linked to the same response. The simplest example of associative learning is the conditioned reflex, discovered by the Russian Institute for Advanced Studies, Jodhpur 59. Unit 7 System Physiology Animal. Physiologist Ivan Pavlov. Pavlov was studying the control of digestive functions in dogs and observed that a dog salivates at the sight or smell of food, a simple autonomic reflex. He discovered that if he rang a bell just before food was presented to the dog, after a few trials the dog would salivate at the sound of the bell, even if no food followed. The salivation reflex was conditioned to be associated with the sound of a bell, a stimulus that normally is unrelated to feeding and digestion. This simple form of learning has been studied extensively in efforts to understand its underlying neuronal mechanisms. In a series of studies led by Richard Thompson, the eye blink reflex of a rabbit in response to a puff of air directed at its eyes was conditioned to be associated with a tone stimulus. After conditioning, the rabbit blinked when it heard the tone. Figure 13. A small and specific area of the cerebellum was discovered to be necessary for this conditioned reflex. Thus, it was possible to localize learning to an identifiable set of synapses in the mammalian brain. Memory. Attempts to treat human neurological diseases have led to localization of areas of the brain involved in the formation and recall of memories. Epilepsy, one SA disorder characterized by uncontrollable increases in neuronal activity in specific parts of the brain. The resulting seizures, or epileptic fits, can endanger the afflicted individual. In the past, serious cases of epilepsy were sometimes treated by destroying the part of the brain from which the surge of activity originated. To find the right area, the surgery was done under local anesthesia, and different regions of the brain were electrically stimulated with electrodes while the patient reported on the resulting sensations. When some regions of association cortex were stimulated, patients reported vivid memories. Such observations were the first evidence that memories have anatomical locations in the brain and exist as properties of neurons and networks of neurons. Yet the destruction of a small area of the brain does not completely erase a memory, so it is postulated that memory is a function distributed over many brain regions and that a memory may be stimulated via many different routes. You can recognize several forms of memory from your own experience. There is immediate memory for events that are happening now. Immediate memory is almost perfectly photographic, but it lasts only seconds. Short-term memory contains less information, but it lasts longer, on the order of 10 to 15 minutes. If you are introduced to a group of new people, you may remember most of their names for 5 or 10 minutes, but you will have forgotten them in an hour or so if you have not repeated them, written them down, or used them in a conversation. Repetition, use, or reinforcement by something that gets your attention, such as the title precedent, facilitates the transfer of short-term memory to long-term memory, which can last for days, months, or years. Knowledge about neuronal mechanisms for the transfer of short-term memory to long-term memory has come from observations of persons who have lost parts of the limbic system, notably the hippocampus. A famous case is that of a man identified as HM, whose hippocampus on both sides of the brain was removed in an effort to control severe epilepsy. Since that surgery, HM has not been able to transfer information to long-term memory. If someone is introduced to him, has a conversation with him, and then leaves the room, when that person returns, he or she is unknown to HM. It is as if the previous conversation had never taken place. HM retains memories of events that happened before his surgery, but he remembers post-surgery events for only 10 or 15 minutes. Memory of people, places, events, and things is called declarative memory because you can consciously recall and describe them. Another type of memory, called procedural memory, cannot be consciously recalled and described. It is the memory of how to perform a motor task. When you learn to ride a bicycle, ski, or use a computer keyboard, you form procedural memories. Although HM is incapable of forming declarative memories, he is capable of forming procedural memories. When taught a motor task day after day, he cannot recall the lessons of the previous day, yet his performance steadily improves. Thus, procedural learning and memory must involve mechanisms different from those used in declarative learning and memory. Our understanding of learning and memory in cellular terms is very rudimentary. New techniques that enable functional imaging of the brain in ways that reveal changes in the metabolic activity of specific regions and structures are greatly enhancing progress in this area. Language abilities are localized in the left cerebral hemisphere. No aspect of brain function is as integrally related to human consciousness and intellect as is language. Therefore, studies of the brain mechanisms that underlie the acquisition and use of language are extremely interesting to neuroscientists. A curious observation about language abilities is that they are usually located in only one cerebral hemisphere, which in 97% of people is the left hemisphere. This phenomenon is referred to as the lateralization of language functions. Some of the most fascinating research on this subject was conducted by Roger Sperry and his colleagues at the California Institute of Technology. Sperry received a Nobel Prize for this work. The two cerebral hemispheres are connected by attractive white matter called the corpus callosum. In one severe form of epilepsy, bursts of action potentials travel from hemisphere to hemisphere across the corpus callosum. Cutting the tract eliminates the problem, and patients function nearly normally following the surgery. But experiments revealed interesting deficits in the language abilities of these split-brain persons. Without the connection between the two hemispheres, the knowledge or experience of the right hemisphere could no longer be expressed in language, nor could language be used to communicate with the right hemisphere. The mechanisms of language in the left hemisphere have been the focus of much research. The experimental subjects are persons who have suffered damage to the left hemisphere and are left with one of many forms of aphasia, a deficit in the ability to use or understand. 60 Institute for Advanced Studies, Jodhpur. 7 Repeating a heard word motor. 14 Language areas of the cortex. Unit 7 System Physiology Animal. Words. These studies have identified several language areas in the left hemisphere. Figure 14. Broca's area, located in the frontal lobe just in front of the primary motor cortex, is essential for speech. Damage to Broca's area results in halting, slow, poorly articulated speech or even complete loss of speech, but the patient can still read and understand language. In the temporal lobe, close to its border with the occipital lobe, one's Wernicke's area, which is more involved with sensory than with motor aspects of language. Damage to Wernicke's area can cause a person to lose the ability to speak sensibly while retaining the abilities to form the sounds of normal speech and to imitate its cadence. Moreover, such a patient cannot understand spoken or written language. Near Wernicke's area is the angular gyrus, which is believed to be essential for integrating spoken and written language. Normal language ability depends on the flow of information among various areas of the left cerebral cortex. Input from spoken language travels from the auditory cortex to Wernicke's area. Figure 14a. Input from written language travels from the visual cortex to the angular gyrus to Wernicke's area. Figure 14b. Beat speaking a written word. Commands to speak are formulated in Wernicke's area and travel to Broca's area and from there to the primary motor cortex. Damage to any one of those areas or the pathways between them can result in aphasia. Using modern methods of functional brain imaging, it is possible to see the metabolic activity in different brain areas when the brain is using language. What is consciousness? This chapter has only scratched the surface of our knowledge about the organization and functions of the human brain, but it may give you some idea of the incredible challenge that neurobiologists face in trying to understand their own brains. Progress is being aided by powerful new technologies such as patch clamping, see figure 10, functional imaging, and neurochemical and molecular methods. However, even these sophisticated new research tools may not allow us to answer the question, what is consciousness? If you see a black dog running across a field, you are conscious of the fact that it is a dog, it is black, and it is a Labrador Retriever. You may remember that the dog's name is Serena, that he belongs to your friend Mira, and that he once 6 years old. From what you have learned in this chapter, imagine how many neurons would be active. Different regions of the left cerebral cortex participate in the processes of repeating a word that is heard and beat speaking a written word. During this experience, neurons in the visual system, the language areas, and in different regions of association cortex. But is being conscious of the black dog simply a result of the fact that all of these neurons are firing at the same time? Your brain is simultaneously processing many other sensory inputs, but you are not necessarily conscious of those inputs. What makes you conscious of the black dog and associated memories and not of other information the brain is processing at the same time? If we could describe all the neurons and all the synapses involved in the conscious experience of seeing and naming a black dog, and then build a computer with devices that modeled all these neurons and connections, would that computer be conscious? It has been said that the question of consciousness results into two types of problems, easy and hard. The easy problems deal with all the cells and circuits that process the information that is involved in conscious experience. The implication of easy is that we seem to have the tools to solve these kinds of problems, as complex as they may be. The hard problems involve explaining how properties of cells and networks result in consciousness, and we seem to lack the proper tools or concepts even to begin to solve these problems. Effectors, making animals move. An animal's central nervous system is more than a processor and storage medium for information. It also allows the animal to respond to information. Effectors are tissues and organs commanded by the CNS to carry out these responses, most of which involve movement. A fascinating array of adaptations enable animals to move. Consider the act of jumping. When you jump, neuronal signals from the motor cortex of your brain are routed through spinal circuits that tell specific leg muscles to contract, extending your legs. Highly skilled and trained athletes can actually outleap their own body height. But as record jumpers go, many other animals, cats, spiders, kangaroos, and fleas, to name just a few, far surpass even the Olympian feats of humans. A flea, for example, can jump more than 200 times its body length. Unlike that of a human jumper, the flea's jumping mechanism does not involve muscles, but works like a slingshot. The flea is so small, and its initial acceleration is so great, that no muscle could contract fast enough to cause such a movement. Instead, at the base of its jumping legs is an elastic material that is compressed by muscles while the flea is resting. When a trigger mechanism is released, the elastic material recoils and fires the flea up and over to its target or away from an enemy. Jumping is just one adaptation an animal can use to respond to information received by its sensory receptors. Effectors also include the internal organs and organ systems that the animal uses to control its internal environment. We will begin this chapter by looking at the mechanisms that power the movement of cilia, flagella, and single cells. The focus of the chapter is on muscles and skeletons, the mechanisms that create mechanical forces and use those forces to change shape and move, and which are the basis for most animal behavior. At the end of the chapter, we will consider a few effectors other than those that create movement. Institute for Advanced Studies, Job for 61. Unit 7 System Physiology Animal. Microtubules, microfilaments, and cell movement. Two components of the cytoskeleton, microtubules and microfilaments, generate cell movement. Both of these structures consist of long protein molecules that can change their length or shape. Microtubules are components of the cytoskeleton. Microtubules are important intracellular effectors for changing cell shape, moving organelles, and enabling cells to respond to their environment. Microtubules generate forces by polymerizing and depolymerizing the protein tubulin. The spindle that moves chromosomes to the meiotic poles of anaphase is made up of microtubules. Another example of microtubule involvement in cell movement is the growth of the axons of neurons in the developing nervous system. Neurons find and make their appropriate connections by sending out long extensions that search for the correct target cells. If polymerization of tubulin is chemically inhibited, these neurons do not extend. Microtubules also generate the small-scale movements of cilia and flagella. Whereas many protists and small invertebrates use cilia for locomotion, larger multicellular animals typically use ciliated cells to move liquids and particles over cell surfaces. Many mollusks, for example, use cilia to circulate a current of water across their gas exchange and feeding surfaces. In humans, the cilia continuously sweep a layer of mucus from deep down in the lungs, up through the windpipe, and into the throat. The mucus carries particles of dirt and dead cells. We can then either swallow or spit out the mucus, and with it, the trapped detritus. Ciliated cells lining the female reproductive tract create currents that sweep eggs from the ovaries into the oviducts and all the way down to the uterus. Flagellated cells maintain a flow of water through the bodies of sponges, bringing in food and oxygen and removing carbon dioxide and wastes. Flagella power the movement of the sperm of most species. Microfilaments change cell shape and cause cell movements. Microfilaments are proteins that change conformation as a means of generating forces. The dominant microfilament in animal cells is the protein actin. Bundles of cross-linked actin strands form important structural components of cells. The microvilli that increase the absorptive surface area of the cells lining the gut are stiffened by actin microfilaments, as are the stereocilia of the sensory hair cells in the mammalian ear. Actin microfilaments can change the shape of a cell by polymerizing and depolymerizing. Microfilaments reach their highest level of organization in muscle cells, which generate large-scale movements. Together with the protein myosin, actin microfilaments generate the contractile forces responsible for many aspects of cell movement and changes in cell shape. The contractile ring that divides an animal cell undergoing mitosis into two daughter cells is composed of actin microfilaments in association with myosin. The mechanisms that many cells employ to engulf materials, endocytosis, also rely on nets of actin and myosin beneath the plasma membrane. Certain cells in multicellular animals travel within the body by amoeboid motion, which is generated by the activity of actin microfilaments and myosin. During development, many cells migrate by amoeboid motion. Throughout an animal's life, phagocytic cells circulate in the blood, squeeze through the walls of the blood vessels, and wander through the tissues by amoeboid motion. The mechanisms of amoeboid motion have been studied extensively in the protist for which this type of movement was named, the amoeba. Amoeboid motion is accomplished by the cell extending a lobe-shaped projection called the pseudopod and then seemingly squeezing itself into that pseudopod. The cytoplasm in the core of the cell is relatively liquid and is called plasmasol, but just beneath the plasma membrane the cytoplasm once much thicker and is called plasmagel. To form a pseudopod, the thick plasmagel in one area of the cell thins, allowing a bulge to form. Just under the cell surface, in the plasmagel, is a network of actin microfilaments that interacts with myosin to squeeze plasmasol into the bulge. As the microfilament network continues to contract, cytoplasm streams in the direction of the pseudopod. When the cytoplasm at the leading edge of the pseudopod converts to plasmagel, the pseudopod stops forming. Thus, the basis for amoeboid motion is the ability of the cytoplasm to cycle through solid gel states and the ability of the microfilament network under the plasma membrane to contract and cause a cytoplasmic stream that pushes out a pseudopod. Muscle contraction. Most behavioral and many physiological responses depend on muscle cells. Muscle cells are specialized for contraction and have high densities of actin and myosin. Muscle cells are found throughout the animal kingdom. Wherever whole tissues contract in animals, muscle cells are responsible. In muscle cells, actin and myosin molecules are organized into microfilaments consisting of two or more molecules. Actin filaments consist of a twisted chain of actin molecules. Myosin filaments are bundles of many myosin molecules. The actin and myosin filaments lie parallel to each other. When contraction is triggered, the actin and myosin filaments slide past each other in a telescoping fashion. This sliding is the mechanism by which muscle cells contract, like those of neurons. Muscle cell plasma membranes can generate action potentials, and it is these action potentials that trigger the contractile machinery. There are three types of vertebrate muscle: smooth muscle, cardiac heart muscle, and skeletal muscle. Figure 15. Although they all use the same contractile mechanism, these three muscle types have important differences that adapt them to their particular functions. Smooth muscle causes slow contractions of many internal organs. Smooth muscle provides the contractile force for most of our internal organs, which are under the control of the autonomic nervous system. Smooth muscle moves food through the digestive tract, controls the flow of blood. 62 Institute for Advanced Studies, Jodhpur. Unit 7 System Physiology Animal. Muscle cell, uterc, aided discs. E. Skeletal muscle. 15 types of vertebrate muscle tissue. The cells of cardiac, or heart, muscle top branch and create a meshwork that resists tearing or breaking. Intercalated discs provide strong mechanical adhesion between the cells. In smooth muscle center, the cells are usually arranged in sheets. Skeletal muscle bottom appears striped or striate. Ed. The individual cells, called muscle fibers, are very large and are multinucleated through blood vessels and empties the urinary bladder. Structurally, smooth muscle cells are the simplest muscle cells. They are usually long and spindle-shaped, and each cell has a single nucleus. They are called smooth because of their microscopic appearance. The actin and myosin filaments in the other types of muscle cells have such a regular arrangement that they give the cells a striped or striated appearance. Such regular arrangement of the filaments does not occur in smooth muscle, hence they appear smooth rather than striated. Figure 15, center. Smooth muscle tissue, such as that from the wall of the digestive tract, has interesting properties. The cells are arranged in sheets, and individual cells in a sheet are in electrical contact with one another through gap junctions. As a result, an action potential generated in the membrane of one smooth muscle cell can spread to all the cells in the sheet of tissue. Thus, the cells in the sheet can contract in a coordinated fashion. Another interesting property of smooth muscle cells is that their plasma membranes are sensitive to being stretched. If the wall of the digestive tract is stretched in one location, as by receiving a swallowed mouthful of food, the membranes of the stretched cells depolarize, reach threshold, and fire action potentials, which cause the cells to contract. Thus, smooth muscle contracts after being stretched, and the harder it is stretched, the stronger the contraction. Other factors that alter the membrane potential of smooth muscle cells are the neurotransmitters of the autonomic nervous system. See figure 9. In the case of the digestive tract, acetylcholine causes smooth muscle cells to depolarize and thus makes them more likely to fire action potentials and contract. Norepinephrine causes these muscle cells to hyperpolarize and therefore makes them less likely to fire action potentials and contract. Figure 16. Cardiac muscle causes the heart to beat. Cardiac muscle looks different from smooth muscle or skeletal muscle when viewed under the microscope. Figure 15, top. Cardiac muscle cells are striated because of the regular arrangement of their actin and myosin filaments. Cardiac muscle cells also branch, and the branches of adjoining cells interdigitate into a meshwork that allows cardiac muscle to resist tearing. As a result, the heart walls can withstand high pressures while pumping blood without the danger of developing leaks. Adding to the strength of cardiac muscle are intercalated discs that provide strong mechanical adhesions between adjacent cells. Like smooth muscle cells, the individual cells in a sheet of cardiac muscle are in electrical contact with one another. Gap junctions in the intercalated discs offer low resistance to ions or electric currents. Therefore, N action potential initiated at one point in the heart spreads rapidly through a large mass of cardiac muscle. Certain tilde specialized cardiac muscle cells, called pacemaker cells, initiate the rhythmic contractions of the heart. Because of these pacemaker cells, the heartbeat is myogenic, generated by the heart muscle itself. The autonomic nervous system modifies the rate of the pacemaker cells, but is not essential for their continued rhythmic function. A heart removed from an animal continues to beat with no input from the nervous system. The myogenic nature of the heartbeat is a major factor in making heart transplants possible. Sliding filaments cause skeletal muscle to contract. Skeletal muscle carries out, or affects, all voluntary movements, such as running or playing a piano, and generates the movements of breathing. Skeletal muscle is also called striated muscle because of its striped appearance. Figure 15, bottom. Skeletal muscle cells, called muscle fibers, are large. Unlike smooth muscle and cardiac muscle cells, each of which has a single nucleus, skeletal muscle fibers have many nuclei because they develop through the fusion of many individual cells. A muscle such as your biceps, which bends your arm, is composed of many muscle fibers bundled together by connective tissue. What is the relation between a skeletal muscle fiber and the actin and myosin filaments responsible for its contraction? Each muscle fiber one is packed with myofibrils, bundles of contractile filaments made up of actin and myosin, figure 17. Within each myofibril are thin actin filaments and thick myosin filaments. If we cut across the myofibril at certain locations, we see only thick filaments. If we cut at other locations, we see only thin filaments. But in most regions of the myofibril, each thick myosin filament is surrounded by 6 thin actin filaments, and conversely, each thin actin filament sits within a triangle of 3 thick myosin filaments. Institute for Advanced Studies, Jodhpur 63. Unit 7 System Physiology Animal. Experiment. Question. What stimulates contraction of smooth muscle? Method. Incubate a strip of smooth intestinal muscle in a saline bath. Measure action potentials and force of contraction. Experiment 1. Stretch intestinal muscle and analyze response. Backslash. Phi muscle membrane potential and L action potentials are recorded. Results. Stretching depolarizes the smooth muscle membrane. The depolarization causes action potentials that activate the contractile mechanism. Experiment 2. Response of muscle strip to neurotransmitters of the autonomic nervous system. Norepinephrine, on the other hand, causes the cells to hyperpolarize, decrease their rate of firing, and decrease their force of contraction. And when acetylcholine is dripped onto the muscle, the cells depolarize, fire action potentials more rapidly, and increase their force of contraction. V. Apply L. Conclusion. Smooth muscle contract. Results. Autonomic neurotransmitters alter membrane resting potential and thereby determine the rate that smooth muscle cells fire action potentials. Conclusion. Smooth muscle contraction is stimulated by stretching by the parasympathetic neurotransmitter acetylcholine. 16 mechanisms of smooth muscle activation. Stretching depolarizes the membrane of smooth muscle cells, and this depolarization causes action potentials that activate the contractile mechanism. The neurotransmitters acetylcholine and norepinephrine also alter the membrane potential of smooth muscle, making it more or less likely to contract. Proteins that help hold the myosin filaments in there. A longitudinal view of a myofibril reveals the reason for regular arrangement. The striated appearance of skeletal muscle and cardiac muscle. The myofibril consists of repeating units called sarcomeres, which are the units of contraction. Each sarcomere once made of overlapping filaments of actin and myosin, which create a distinct band pattern. As the muscle contracts, the sarcomeres shorten, and the appearance of the band pattern changes. The observation that the widths of the bands in the sarcomeres change when a muscle contracts led two British biologists, Hugh Huxley and Andrew Huxley, to propose a molecular mechanism of muscle contraction. Let's look at the band pattern of a sarcomere in detail. Each sarcomere is bounded by Z lines, which are structures that anchor the thin actin filaments. Centered in the sarcomere is the A band, which contains all the myosin filaments. The H zone and the I band, which appear light, or regions where actin and myosin filaments do not overlap in the relaxed muscle. The dark stripe within the H zone is called the M band. It contains the bundles of myosin filaments are held in a centered position within the sarcomere by a protein called titin. Titin is probably the longest polypeptide in the body. It runs the full length of the sarcomere from Z line to Z line, and each titin molecule runs right through a myosin bundle. Between the ends of the myosin bundles and the Z lines, titin molecules have the properties of a bungee cord. They are very stretchable. In a relaxed skeletal muscle, resistance to stretch is mostly due to the elasticity of the titin molecules. When the muscle contracts, the sarcomere shortens. The H zone and the I band become much narrower, and the Z lines move toward the I band as if the actin filaments were sliding into the region occupied by the myosin filaments. This observation led Huxley and Huxley to propose the sliding filament theory of muscle contraction. Actin and myosin filaments slide past each other as the muscle contracts. 64 Institute for Advanced Studies, Jodhpur. Unit 7 System Physiology Animal. Each muscle fiber is a multinucleate cell containing numerous myofibrils, which are highly ordered assemblages of thick myosin and thin actin filaments. The structure of the myofibrils gives muscle fibers their characteristic striated appearance. Actin, myosin interactions cause filaments to slide. To understand what makes the filaments slide, we must examine the structures of actin and myosin, figure 18. Each myosin molecule consists of two long polypeptide chains coiled together, each ending in a large globular head. A myosin filament is made up of many myosin molecules arranged in parallel, with their heads projecting laterally from one or the other end of the filament. An actin filament consists of a helical arrangement of two chains of actin monomers twisted together like two strands of pearls. Twisting around the actin chains is another protein, tropomyosin, and attached to it intervals are molecules of troponin. We'll discuss the roles of these last two proteins in the following section. The myosin heads have sites that combine to actin and thereby form cross bridges between the myosin and the actin filaments. The myosin heads also have ATPase activity, that is, they bind and hydrolyze ATP. The energy released when this happens changes the conformation, and therefore the orientation, of the myosin head. Together, these details explain the cycle of events that cause the actin and myosin filaments to slide past each other and shorten the sarcomere. A myosin head binds to an actin filament, see figure 20. Upon binding, the head changes its orientation with respect to the myosin filament, thus exerting a force that causes the actin filament to slide about 5 to 10 nanometers relative to the myosin filament. Next, the myosin head binds a molecule of ATP, which causes it to release the actin. When the ATP is hydrolyzed, the energy release causes the myosin head to return to its original conformation, in which it can bind again to actin. It is as if the energy from ATP hydrolysis is being used to cock the hammer of a pistol, and contact of the myosin head with an actin binding site pulls the trigger. We have been discussing the cycle of contraction in terms of a single myosin head. Don't forget that each myosin filament has many myosin heads at both ends and is surrounded by 6 actin filaments. Thus, the contraction of the sarcomere involves a great many cycles of interaction between actin and myosin molecules. That is why when a single myosin head breaks its contact with actin, the actin filaments do not slip backward. Institute for Advanced Studies, Jodhpur 65. Unit 7 System Physiology Animal. Troponin has 3 subunits. One binds actin, one binds tropomyosin, and one binds Ca. Actin monomer tropomyosin troponin myosin filament actin filament. Globular. Head, underscore and myosin molecule. Linear. 18 polypeptide chain actin and myosin filaments overlap to form myofibrils. Myosin filaments are bundles of molecules with globular heads and polypeptide tails. Actin filaments consist of two chains of actin monomers twisted together. They are wrapped by chains of the polypeptide tropomyosin and studded at intervals with another protein, troponin. An interesting aspect of this contractile mechanism is that ATP is needed to break the actin myosin bonds, but not to form them. Thus muscles require ATP to stop throughout the cytoplasm of the muscle fiber, also called the sarcoplasm, figure 19. Motor neuron, underscore contracting. This fact explains why muscles stiffen soon after animals die, a condition known as rigor mortis. Death stops the replenishment of the ATP stores of muscle cells, so the actin myosin bonds cannot be broken, and the muscles stiffen. Eventually the proteins begin to lose their integrity, and the muscles soften. These events have regular time courses that differ somewhat for different regions of the body. Therefore, an examination of the stiffness of the muscles of a corpse can help a coroner estimate the time of death. Forward muscle fiber. L and action potential, black arrows, arrives at the meotor neuron terminal. FY the muscle fiber plasma membrane generates an action potential that spreads down T tubules. Muscle contractions are initiated by action potentials from motor neurons arriving at the neuromuscular junction. The axons of motor neurons are generally highly branched and can synapse with up to 100. The axons of motor neurons are generally highly branched and can synapse with up to 100. Excitable, that is, their plasma membranes can generate and conduct action potentials. In the case of skeletal muscle fibers, but not smooth or cardiac muscle fibers, all action potentials are initiated by motor neurons. When an action potential arises at the neuromuscular junction, the neurotransmitter acetylcholine is released from the motor neuron, diffuses across the synaptic cleft, binds to receptors in the postsynaptic membrane, and causes ion channels in the motor end plate to open. Most of the ions that flow through these channels are not plus, and therefore the motor end plate is depolarized. The depolarization spreads to the surrounding plasma membrane of the muscle fiber, which contains voltage-gated sodium channels. When threshold is reached, the plasma membrane fires an action potential that is conducted rapidly to all points on the surface of the muscle fiber. An action potential in a muscle fiber also travels deep within the cell. The plasma membrane is continuous with a system of tubules that descends into and branches. Plasma. Sarcoplasmic. 19 reticulum. T-tubules in action and action potential at the neuromuscular junction. Spreads throughout the muscle fiber via a network of T-tubules, triggering the release of Ca from the sarcoplasmic reticulum. The action potential that spreads over the plasma membrane also spreads through this system of transverse tubules, or T-tubules. The T-tubules come very close to a network of intracellular membranes called the sarcoplasmic reticulum. The sarcoplasmic reticulum forms a membrane-enclosed compartment that surrounds every myofibril. Calcium pumps in the sarcoplasmic reticulum cause it to take up Ca ions from the sarcoplasm. Therefore, when the muscle fiber is at rest, there is a high concentration of Ca in the sarcoplasmic reticulum and a low concentration of Ca in the sarcoplasm. Spanning the space between the membranes of the T-tubules and the membranes of the sarcoplasmic reticulum are two proteins. One protein, which is located in the T-tubule membrane, is voltage sensitive and changes its conformation when an action potential reaches it. The other protein is located in the sarcoplasmic reticulum membrane and is a Ca greater than channel. 66 Institute for Advanced Studies, Jodhpur. Membrane. Unit 7 System Physiology Animal. When it is activated by an action potential, the voltage-sensitive protein opens the Ca channel, and Ca ions diffuse out of the sarcoplasmic reticulum and into the sarcoplasm surrounding the actin and myosin filaments. It is these Ca ions that trigger the interaction of actin and myosin and the sliding of the filaments. How do the Ca ions do this? An actin filament, as we have seen, is a helical arrangement of two strands of actin monomers. Lying in the grooves between the two actin strands is a two-stranded protein tropomyosin. See figure 18. At regular intervals, the filament also includes a globular protein, troponin. The troponin molecule has three subunits. One binds actin, one binds tropomyosin, and one binds Ca. When Ca is sequestered in the sarcoplasmic reticulum, the tropomyosin strands block the sites on the actin filament where myosin heads combined. When the T-tubule system depolarizes, Ca is released into the sarcoplasm, where it binds to troponin, changing its conformation. Because the troponin is bound to the tropomyosin, this conformational change of the troponin twists the tropomyosin enough to expose the actin, myosin binding sites. Thus the cycle of making and breaking actin, myosin bonds is initiated, the filaments are pulled past each other, and the muscle fiber contracts. When the T-tubule system repolarizes, the calcium pumps remove the Ca ions from the sarcoplasm, causing the tropomyosin to return to the position in which it blocks the binding of myosin heads to actin, and the muscle fiber returns to its resting condition. Figure 20 summarizes this cycle. RF exposes actin myosin binding sites. As long as binding sites and ATP are available TH, the cycle of actin and myosin interactions continues, and the filaments slide past each other. Calmodulin mediates Ca greater than control of contraction in smooth muscle. Smooth muscle cells do not have the troponin, tropomyosin mechanism for controlling contraction, but Ca2+ still plays a critical role. A Ca2 inches influx into the sarcoplasm of a smooth muscle cell can be stimulated by action potentials, by hormones, or by stretching. The Ca that enters the sarcoplasm combines with a protein called calmodulin. The calmodulin, Ca, complex activates an enzyme called myosin kinase, which can phosphorylate myosin heads. When the myosin heads in smooth muscle are phosphorylated, they can undergo cycles of binding and releasing actin, causing muscle contraction. And releasing actin, causing muscle contraction. As Ca2+ is removed from the sarcoplasm, the myosin heads bind to actin, ATP is released. F underscore. In the power stroke. The action potential that spreads over the plasma membrane also spreads through this system of transverse tubules, or T-tubules. The T-tubules come very close to a network of intracellular membranes called the sarcoplasmic reticulum. The sarcoplasmic reticulum forms a membrane-enclosed compartment that surrounds every myofibril. Calcium pumps in the sarcoplasmic reticulum cause it to take up Ca ions from the sarcoplasm. Therefore, when the muscle fiber is at rest, there is a high concentration of Ca in the sarcoplasmic reticulum and a low concentration of Ca in the sarcoplasm. Spanning the space between the membranes of the T-tubules and the membranes of the sarcoplasmic reticulum are two proteins. One protein, which is located in the T-tubule membrane, is voltage sensitive and changes its conformation when an action potential reaches it. The other protein is located in the sarcoplasmic reticulum membrane and is a Ca greater than channel. 66 Institute for Advanced Studies, Jodhpur. Membrane. Unit 7 System Physiology Animal. When it is activated by an action potential, the voltage-sensitive protein opens the Ca channel, and Ca ions diffuse out of the sarcoplasmic reticulum and into the sarcoplasm surrounding the actin and myosin filaments. It is these Ca ions that trigger the interaction of actin and myosin and the sliding of the filaments. How do the Ca ions do this? An actin filament, as we have seen, is a helical arrangement of two strands of actin monomers. Lying in the grooves between the two actin strands is a two-stranded protein tropomyosin. See figure 18. At regular intervals, the filament also includes a globular protein, troponin. The troponin molecule has three subunits. One binds actin, one binds tropomyosin, and one binds Ca. When Ca is sequestered in the sarcoplasmic reticulum, the tropomyosin strands block the sites on the actin filament where myosin heads combined. When the T-tubule system depolarizes, Ca is released into the sarcoplasm, where it binds to troponin, changing its conformation. Because the troponin is bound to the tropomyosin, this conformational change of the troponin twists the tropomyosin enough to expose the actin, myosin binding sites. Thus the cycle of making and breaking actin, myosin bonds is initiated, the filaments are pulled past each other, and the muscle fiber contracts. When the T-tubule system repolarizes, the calcium pumps remove the Ca ions from the sarcoplasm, causing the tropomyosin to return to the position in which it blocks the binding of myosin heads to actin, and the muscle fiber returns to its resting condition. Figure 20 summarizes this cycle. RSSRRY. RCA is released from the sarcoplasmic reticulum. Tropomyosin actin filament troponin. If Ca is returned to the sarcoplasmic reticulum, the muscle relaxes. ATP is hydrolyzed and the myosin head returns to its resting conformation. Ca in the sarcoplasm exposes actin myosin binding sites. As long as binding sites and ATP are available TH, the cycle of actin and myosin interactions continues, and the filaments slide past each other. Calmodulin mediates Ca greater than control of contraction in smooth muscle. Smooth muscle cells do not have the troponin, tropomyosin mechanism for controlling contraction, but CA2+ still plays a critical role. A CA2 inches influx into the sarcoplasm of a smooth muscle cell can be stimulated by action potentials, by hormones, or by stretching. The CA that enters the sarcoplasm combines with a protein called calmodulin. The calmodulin, CA, complex activates an enzyme called myosin kinase, which can phosphorylate myosin heads. When the myosin heads in smooth muscle are phosphorylated, they can undergo cycles of binding and releasing actin, causing muscle contraction. As CA2+ is removed from the sarcoplasm, the myosin heads bind to actin, ATP is released. F underscore. In the power stroke, the myosin head changes conformation, filaments slide past one another. Dissociates from calmodulin, and the activity of myosin kinase falls. In addition, another enzyme, myosin phosphatase, dephosphorylates the myosin and helps stop the actin-myosin interactions. Single skeletal muscle twitches are some integrated contractions. In skeletal muscle, the arrival of an action potential at a neuromuscular junction causes an action potential in a muscle fiber. The spread of that action potential through the T-tubule system of the muscle fiber causes a minimum unit of contraction, called a twitch, at which can be measured in terms of the tension, or force, it generates figure 21a, a single action potential. Institute for Advanced Studies, Jodhpur 67. A. Force. Unit 7 System Physiology Animal. Stimulates a single twitch, but the ultimate force generated by a muscle can vary enormously depending on how many muscle fibers are in its motor units. In muscles responsible for fine movements, such as those of the fingers, a motor neuron may innervate only one or a few muscle fibers, but in a muscle that produces large forces, such as the biceps, a motor neuron innervates a large number of muscle fibers. Still, however, at the level of the single muscle fiber, a single action potential stimulates a single twitch. If action potentials reaching the muscle fiber are adequately separated in time, each twitch is a discrete, all or none phenomenon. If action potentials are fired more rapidly, however, new twitches are triggered before the myofibrils have had a chance to return to their resting condition. As a result, the twitch is sum, and the tension generated by the fiber increases and becomes more sustained. Thus, an individual muscle fiber can show greater response to increased levels of stimulation by its motor neuron. At high levels of stimulation, the calcium pumps in the sarcoplasmic reticulum can no longer remove CA ions from the sarcoplasm between action potentials, and the contractile machinery generates maximum tension, a condition known as tetanus figure 21b. Do not confuse this condition with the disease tetanus, which is caused by a bacterial toxin and is characterized by spastic contractions of skeletal muscles. At high levels of stimulation, the calcium pumps in the sarcoplasmic reticulum can no longer remove CA ions from the sarcoplasm between action potentials, and the contractile machinery generates maximum tension, a condition known as tetanus figure 21b. Do not confuse this condition with the disease tetanus. At high levels of stimulation, the calcium pumps in the sarcoplasmic reticulum can no longer remove CA ions from the sarcoplasm between action potentials, and the contractile machinery generates maximum tension, a condition known as tetanus figure 21b. Do not confuse this condition with the disease tetanus, which is caused by a bacterial toxin and is characterized by spastic contractions of skeletal muscles. How long a muscle fiber can maintain a tetanic contraction depends on its supply of ATP. Eventually, the fiber will become fatigued. It may seem paradoxical that the lack of ATP causes fatigue, since the action of ATP is to break actin, myosin bonds. But remember that the energy released from the hydrolysis of ATP recox the myosin heads, allowing them to cycle through another power stroke. When a muscle is contracting against a load, the cycle of making and breaking actin, myosin bonds must continue to prevent the load from stretching the muscle. The situation is like rowing a boat upstream. You cannot maintain your position relative to the stream bank by just holding the oars out against the current. You have to keep rowing. Likewise, actin, myosin bonds have to keep cycling to maintain tension in the muscle. The level of tension generated by a muscle depends on how many motor units in that muscle are activated. Whether a muscle contraction is strong or weak depends both on how many of the motor neurons that synapse with that muscle are firing and on the rate at which those neurons are firing. These two factors can be thought of as spatial summation and temporal summation, respectively. Unit of contraction of a muscle fiber. Have a summed effect. J. A stimulus elicits a twitch. The minimum go twitch is in quick, Uchi, ion. Stimulus, R. 0. Force. TTT muscle at S relax when stimulation stops, J. 7. 8 some twitches bring the tetanus sustained by a muscle fiber to maximum contraction, high rate of stimulation. Known as tetanus. PAL. Twitches and tetanus, action potentials from a motor neuron cause a muscle fiber to twitch. Sinus odd FFBBB periodine. Twitches in quick succession can be summed. Beat summation of many twitches can bring the muscle fiber to the maximum level of contraction, known as tetanus. Many muscles of the body maintain a low level of tension even when the body is at rest. For example, the muscles of the neck, trunk, and limbs that maintain our posture against the pull of gravity are always working, even when we are standing or sitting still. Muscle tone comes from the activity of a small but changing number of motor units in a muscle. At any one time, some of the muscle's fibers are contracting and others are relaxed. Muscle tone is constantly being readjusted by the nervous system. Muscle strength and performance. Muscle fiber types determine endurance and strength. Not all skeletal muscle fibers are alike, and a single muscle contains more than one type of fiber. The two major types of skeletal muscle fibers differ in the properties of their myosin molecules, and these myosin variants have different rates of ATPase activity. Those with high ATPase activity can recycle their actin, myosin cross bridges rapidly and are therefore called fast twitch fibers. Slow twitch fibers have lower ATPase activity, so they can develop tension more slowly and spread maintain it over a longer period of time. Slow twitch fibers are also called red muscle because they contain lots of the oxygen-binding molecule myoglobin. They have many mitochondria, and they are well supplied with blood vessels. These characteristics increase their capacity for oxidative metabolism. The maximum tension a slow twitch fiber produces is low and develops slowly, but it is highly resistant to fatigue. Because slow twitch fibers have substantial reserves of fuel, glycogen and fat, their abundant mitochondria can maintain steady, prolonged production of ATP as long as oxygen is available. Muscles with high proportions of slow twitch fibers are good for long-term aerobic work, that is, work that requires lots of oxygen. Champion. Long-distance runners, cross-country skiers, swimmers, bicyclists, and other athletes whose activities require endurance have leg and arm muscles consisting mostly of slow twitch fibers. Fast twitch fibers are also called white muscle because, in comparison to slow twitch fibers, they have fewer mitochondria, little or no myoglobin, and fewer blood vessels. The white meat of domestic chickens has a high percentage of fast twitch fibers. Fast twitch fibers can develop maximum tension more rapidly than slow twitch fibers can, and that maximum tension is greater, but fast twitch fibers fatigue rapidly. The myosin of fast twitch fibers can put the energy of ATP to work very rapidly, but the fibers cannot replenish ATP quickly enough to sustain contraction for a long time. Fast twitch fibers are especially good for short-term work that requires maximum strength. Champion weightlifters and sprinters have leg and arm muscles with high proportions of fast twitch fibers. What determines the proportion of fast and slow twitch fibers in your skeletal muscles? The most important factor is genetic heritage, so there is some truth to the statement that champions are born, not made. To a certain extent, you can alter the properties of your muscle fibers through training. But a person born with a high proportion of fast twitch fibers will never become a champion marathon runner, and one born with a high 68 Institute for Advanced Studies, Jodhpur. What determines the proportion of fast and slow twitch fibers in your skeletal muscles? The most important factor is genetic heritage, so there is some truth to the statement that champions are born, not made. To a certain extent, you can alter the properties of your muscle fibers through training. But a person born with a high proportion of fast twitch fibers will never become a champion marathon runner, and one born with a high 68 Institute for Advanced Studies, Jodhpur. Unit 7 System Physiology Animal. Proportion of slow twitch fibers will never become a champion sprinter. The strength of a muscle fiber is related to its length. Have you ever done a pull-up? If you have, you know that two parts of this exercise are especially difficult. When you are hanging from the bar with your arms fully extended, it is hard to get the pull-up started, and when your chin is just about to the bar, pulling yourself up the last small distance is difficult. These experiences are explained by the structure of the sarcomere. When a muscle is stretched and the sarcomeres are lengthened, there is less overlap between the actin and myosin filaments, therefore, fewer cross bridges can form, and less force can be produced. In fact, if the sarcomeres are stretched too much, there is no overlap between the actin and myosin, and no force can be produced. How would a muscle recover from such a difficult situation? The bungee cord-like tight molecules create enough elastic recoil to pull the actin and myosin fibrils back into an overlapping arrangement. When the muscle is fully contracted, the actin and myosin filaments overlap so much that the myosin bundles are pressed up against the Z lines. Because they have no place to go, additional shortening is difficult. You can see the relationship between the length of a muscle fiber and its ability to develop tension in figure 22. Myosin actin. 22. Strength and length. The amount of force a sarcomere can generate depends on its resting length. When a muscle is stretched, the sarcomeres lengthen. There is less overlap between the actin and myosin filaments, and less force is produced. Overstretched sarcomeres produce no force because there is no overlap between the actin and myosin. Exercise increases muscle strength and endurance. Different types of exercise produce different physical conditioning responses. In general, anaerobic activities, such as weightlifting, increase strength, and aerobic activities, such as jogging, increase endurance. What is the physiological basis for these differences? Strength is quite simply a function of the cross-sectional area of muscles. The more actin and myosin filaments in a muscle or a muscle fiber, the more tension it can produce. When athletes undertake strength training, they use weights or exercises such as push-ups to repeatedly contract specific muscles under heavy loads. Repetitions are usually done until the muscle is completely fatigued. Such stress on a muscle probably does minor tissue damage, hence the soreness a day after a hard workout, but it also induces the formation of new actin and myosin filaments in existing muscle fibers. The muscle fibers, and hence the muscles, get bigger and stronger. In extreme cases, and after serious muscle damage, new muscle fibers can also be produced from stem cells called satellite cells in the muscle. In general, however, the major effect of strength training is to produce bigger, rather than more, muscle fibers. Aerobic exercise has a completely different effect on muscles. It enhances their oxidative capacity. This effect comes from increases in the number of mitochondria, increases in enzymes involved in energy utilization, and an increase in the density of capillaries that deliver oxygen to the muscle. There is also an increase in myoglobin, which facilitates the diffusion of oxygen throughout the muscle fibers and provides a store of oxygen for use when oxygen delivery by the blood is insufficient. In this way, aerobic training can stimulate many fast twitch fibers to increase their oxidative capacity. Muscle fuel supply limits performance. Muscles have three systems for obtaining the ATP they need for contraction. The immediate system, preformed ATP, and creatine phosphate O, the glycolytic system, metabolizing carbohydrates to lactate, and pyruvate O, the oxidative system, metabolizing carbohydrates or fats all the way to HO and CO. The capacity of these three systems and the rates at which they can produce ATP determine both work capacity and endurance. Figure 23. ATP is stored in muscles in very small amounts. However, muscle fibers also contain a storage compound called creatine phosphate (CP). This molecule stores energy in a phosphate bond, which it can transfer to ADP. The total energy available in all the muscles of your body in the form of ATP and CP, the immediate energy system, is only about 10 calories. However, it is available immediately, and it enables fast twitch fibers to generate a lot of force quickly. The immediate system is exhausted in only a few seconds. The glycolytic system is able to come online within a few seconds to replace the ATP depleted at the onset of muscle activity. The glycolytic enzymes are located in the cytoplasm of the muscle fiber, and therefore the ATP they generate is rapidly available to the myosin filaments. However, glycolysis alone is an inefficient way to produce ATP, and it rapidly leads to the accumulation of lactic acid, which slows the process. Institute for Advanced Studies, Jodhpur 69. Unit 7 System.